Hey guys. So sorry I haven't made videos in a while. It's Monday and it's already been a long week. For those of you who don't know, I'm a swimmer and I didn't bring sneakers to the gym today, so I came home wearing heels with my sweatpants. So sorry I haven't filmed in a while, just want to let you know that it's been a long week today. So in 2002, why am I starting? This is chapter 14. This is section 1. I will be doing section 2 tomorrow. Hopefully we'll be able to be done with chapter 14 in a few days because I have a test on Friday. So for all those who watch in my class, I hope to get this done for Friday for you guys, so you guys all know the material. So let's get started. In 1902, Archibald Garrett thought about genes and their role in the cell. As a result, he hypothesized that genes dictate the phenotypes through enzymes that catalyze specific chemical reactions. Specifically, he believed that symptoms of a disease were the result of a person's inability to make a specific enzyme. Now, Garrett was the first person to postulate this, but it wasn't researched until much later. And when I say much later, I mean the 1940s. So, in 1941, American geneticists George Beadle and Edward Tatum. Beadle and Tatum. Sounds like a buddy cop movie, doesn't it? So, Beadle and Tatum tried to demonstrate the relationship between genes and enzymes. They did this by inflicting gene mutations on the bread mold Neurospora crassa using X-rays. The cells were placed in a minimal medium. The minimal medium is a medium that contains the most basic nutrients in order for this mold to grow. So basically it contained inorganic salts, glucose, and the vitamin biotin. That's basically like bread and water for us. So now this is how the experiment worked. So here they have their mold. They zap them with X-rays and they put them in the minimal media. So from there, they put them into different categories, which you can actually look this experiment up online. I'm not going to go into depth with it, but it's not that difficult to understand. So what they did was they added, so basically they wanted to see which gene was activated, gene A, gene B, or gene C, because throughout this whole thing, eventually with these genes, it creates arginine. So through this process of creating, the precursor becomes ornithine through enzyme A, which is turned on by gene A. So they were trying to figure out which one was deactivated. So if gene A was deactivated, there would be no growth in the minimal medium. But if I added ornithine, then there would be growth. And same thing for gene B. If I deactivated gene B but then added citrulline, then there would be growth. So they were just trying to figure that out. So look that up online because I'm not going to go into depth with it if you don't understand. Through this, Beadle and Tatum were able to prove the one gene, one enzyme theory. So with further research, scientists were able to understand that genes do not only code for enzymes, but also for proteins. And since enzymes are proteins, they were able to modify this to one gene, one protein. It was specified even more when scientists realized that more than one gene could code for a protein. So in other words, a gene codes for an individual polypeptide, which makes it protein. So the theory now stands as one gene, one polypeptide. So if you didn't understand that last paragraph, just know one gene, one polypeptide. But how do genes code for a polypeptide? Now this is where RNA comes in. RNA is a very important nucleic acid. It's a nucleic acid that is similar to DNA. Now, let's examine some of the differences.
So in one corner, here we have RNA. RNA is a single-stranded nucleic acid with 4 nitrogenous bases: adenine, guanine, cytosine, and uracil. Its backbone is also made up of ribose, a sugar. Now in the other corner, we have DNA, a double helix that is also made up of 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. Its backbone is made up of another sugar, deoxyribose.
Transcription and translation are the two phases that are necessary for protein synthesis. So basically, nucleic acids and proteins create information written in two separate languages, and in order for it to be read, these two processes have to occur. Transcription is the synthesis of RNA, using information in the DNA. Basically, it is the process of transcribing the information of DNA onto an RNA molecule. This RNA molecule is known as messenger RNA, or simply mRNA. It is called messenger RNA because it carries a genetic message from the DNA into the cytoplasm where protein synthesis occurs. Yay! Okay, in translation, the code is transcribed onto the mRNA as read, and polypeptides are synthesized. Translation occurs when ribosomes, particles that are able to facilitate the orderly linking of amino acids into polypeptide chains. But don't worry, we'll go into the specifics of translation in one of my upcoming videos. So this process, transcription and translation, being the gap, between DNA and proteins, is considered to be the central dogma of DNA. And as you can see behind me, it all comes together. So your DNA goes to RNA, and RNA goes to protein. So the process of DNA to RNA is transcription, and RNA to protein is translation. When scientists first realized that DNA was the genetic code, they wanted to know how many codons corresponded with an amino acid. They knew that there were 20 amino acids, and in order to satisfy this, scientists hypothesized that the genetic code is read in groups of 3 bases. These triplets of base pairs are known as triplet codes, and the base pairs that make up the triplet codes are known as codons. In total, there are 64 possible combinations of codons. 3 out of those 64 codons terminate protein synthesis and release the polypeptide from the translation machinery. These triplets are known as stop codons, which we'll talk about later. We'll name specific stop codons in the later video of translation. Another important triplet codon is AUG. AUG serves as the start codon and initiates translation. The location of AUG sets the reading frame. A reading frame is the correct way in which triplets of codons are viewed. So our textbook gives a really good example of this. So the phrase, "the red dog ate the bug," makes sense to us. But if you shift the reading frame over by 1 letter, you start at the wrong point and it becomes, I don't even know how to read this, it's crazy. Go tet heb ug. Sounds like complete gibberish, but it's crazy how much 1 frame of letters can do. So yeah, that's why it matters that your everything is read properly in translation, so proper proteins can be synthesized, so you don't turn out to be a wreck. But that's it. So tomorrow I'm going to go in depth about transcription and then hopefully translation. So yeah, have a nice night.